Here we go. First of all, thank you so much, Aubrey and Martin, for the invitation. I'm really excited to be here talking about what we do in my lab. So let's start with some basic stuff. And you know better than anybody else that during the last 150 years, basically we were able to do different things to increase our lifespan. So it went from 40s to 80s. However, during the same amount of time, nothing really happened when it comes to reproductive lifespan. So no matter what you do, no matter what supplement you take, no matter what happens, menopause will come and knock your door at 51 or 52 in the best case scenario. And the problem with menopause is not just like what we used to think about it. That is like stop having babies, nothing happens. Now we know there are like many, many things happening that they can involve or affect every single layer of your body. It could be something like small like a wrinkle, but it goes deeper and deeper and deeper, causing muscle atrophy, affects the bone, as you know, increase the risk of osteoporosis, cardiovascular diseases, immunological diseases, also increased risk of cancer, and finally, cognitive function. So the big question here is like why this is happening. And Mother Nature sometimes is really bad with females, many times to be sincere. And there's something called in reproduction called the dogma of reproductive biology that is really simple and really old, but also kind of tragic. And what it says is that the number of eggs or germ cells that you have in your ovary is all what you're gonna have. And from there, everything goes down. And this is based in the biology or how the ovary is developed. So let's use this example. This is human. Here we have weeks. So the primordial germ cells are the cells that will be originating all the germ cells. So we have here, then they go to a massive mitotic cell division to reach a number of, best case scenario, 7 million. During that process, they're going to start to differentiate and go and start meiosis, folliculogenesis, and this will be involving that there's a drop, a massive drop in the number of germ cells. That, if everything goes well, it will turn into 1 million at birth. But the thing doesn't stop there, because by the time of your first menstruation or menses, this is 400,000. And keeps going down and going down up to the moment that menopause comes to your door, 51, 52, and where technically you should have like 1,000. During the last few years, there's a controversy about there's stem cells, adult stem cells in many organs, right? So why not in the ovary? So this controversy is really like two teams. The team of ovarian pessimists, that they say that there's no kind of any cells that could be increasing the ovarian reserve, and then the ovarian optimist that they say that actually can happen. The true story is that it doesn't matter in what team you are, those cells are doing nothing, because at the end of the story, you have the same effect. However, to every single rule, there is an exception. And let me introduce you this lovely girl. She is the naked Morat. Probably she's not the most beautiful girl in town, but this girl already won in life. So she's originally from Africa, from the region of Kenya, Ethiopia, and Somalia. And she's part of the African moles. She is really famous because she is a social mammal. Basically, there are two social mammals, and she is the most important or most famous one. This means that this animal, just like bees or ants, they have a queen, and then other set of animals that are in charge of other functions of the colony. As you can imagine, this girl is the only one that has babies, and also they have a long gestation, but also they can be fertile, or they can induce as a queen at 6 months. So this is different compared to what is happening with bees or ants, where they're primed to be a queen. In the naked Morat world, every girl can be a queen if you remove them from the colony. So that means that the mechanism that is happening there, we can trick with it. In the field of aging, it has been really famous because this little bug that is the size of a Twinkie and the color of a Twinkie, and almost with not expiration date like the Twinkie, can live like up to 37 years. Now we know that also it can be reached 41 in certain labs. Also, it's famous because they have a mutation that, let's say, that allows them to feel the pain in a different way. They can live without oxygen up to 18 minutes and they're happy. They're really highly, highly resistant to cancer. But we're talking about reproductive aging, so why do I care about the naked mole rat? And the reason is that these girls can be having babies during 41 years. They have no decline in fertility during their time lifespan. So the big question here is, how this little bug that is so weird and not that pretty can do this? And that is something that we were working in my lab to try to answer. So many possibilities, but we decided to reduce to 3 main questions. Maybe they have tons and tons of germ cells, so they just don't use it all. The second one is like maybe instead that have that decline of germ cells, they don't kill as many. And the third one is that maybe they're putting more germ cells into the pocket. However, it sounds quite simple, get the ovaries, analyze them, but we didn't have a roadmap for naked mole rats because we have it for mouse, we have for human, but not for this animal. So we decided, okay, it's a rodent, let's try to use the map of the naked mole rat, of the mouse. And well, for that, let's think about it, how this works. So in the mouse, we have like the specification of the primordial germ cells or germ cell line really early during the development. Later on, they will be moving. They migrate into the gonad. During this time, they go into like a repress of the somatic program. They will go to the gonad. They start like sex differentiation. There's got to massive change in DNA methylation. And around 13.5, they get into meiosis. After that, they start like doing the follicle formation. They have these clusters first of all the oocytes and then start to breaking apart. And around day 22, they have follicles in the way that we think. So that is what we expect. But actually, the important part is that we didn't see anything like that. What we saw, in fact, in the naked mole rat was more a situation where there were tons and tons and tons and tons of cells that we didn't really know what they were. And it's not up to 3 months after birth when we start to see some follicles in the way that we think about follicles. So the question here is like, what are the cells that are the postnatal day 1, 5, etc.? What are they? So to try to know what is this, we decided to use a marker of germ cells that is really conserved across the species. This is the DDX4 or BASA that probably you heard before. And the cool part is that actually work in the naked mole rat. And what we saw is that, come on, the entire ovary are basically germ cells. This is a postnatal day 1. Everything that you see in red are germ cells. And this doesn't stop there. It keeps going at P5, P8, P15, P28, and up to 3 months after is when it starts to look a little bit like mouse. But it's still like not like mouse because let's make a close-up of this little region. And we have many cells here. So if we remove the red, in the mouse I said that it's like step 1, step 2, step 3, step 4. And what are we seeing here is that in the case of the naked mole rat, it's more like kind of all mixed together. So we have early stages, then we have middle stages, and a little bit of later stages. And that's why we have nest, primary, primordial, and secondary. We have tons and tons of germ cells, right? But not working. It's not working. Hello? Okay, so we have, thank you. We have tons and tons of germ cells, but that doesn't work for a reviewer, right? So we actually count them. And now we know that in fact, it's not working. Okay. Okay. Meanwhile, we are trying to figure out our technical problems. The reviewers don't like to say tons and tons, so actually we count them. And what we found is that there are like a big number of germ cells in the naked mole rat that we skip the graph, but well, we can go back in a little bit later. And this is 95 times, oh, here we go. we have like 1.5 million of germ cells in the naked mole rat at 8 days after birth. This is equivalent to what are we seeing with human. So this is 95 times bigger or greater than what are we seeing in mouse. But also another important part of this is that it's asynchronic like in human. So we have all this situation. So now that we know that we have a lot, we need to know what is the timeline of this. So to do that, we want and think about, okay, what is the timeline for the germ cell development? And what we think about is like, let's go for markers that are highly conserved across the species. And then we decide to look for really early germ cells that are primordial germ cells that are positive for pluripotency factors, plus LIMP1. And actually what we saw is that, in fact, we have cells in the ovary that are positive for pluripotency factors from P1 to P90. Yes, we have a peak here, but the important part is that even when they reduce, they never disappear. This is OC4, but it's also the same thing for SOX2 and another markers of pluripotency. And at the same time for LIMP1. So they are positive for germ cell markers and positive for pluripotency and PGCs. Again, here we have a peak, they drop, but they never disappear. LIMP1, same situation. So now we know that we have these cells. We also think about what are the rest of cells that are there, because clearly they were forming clusters. So it's like they're like before, after, what is happening. So we decide to analyze the entry into meiosis using STRA8. This marker is saying like, okay, cells start to go into meiosis, and then we saw that there's nothing at the beginning, a little bit, and then again, goes up, drops, and is still present there. So meaning that probably they're like cells that are constantly entering into meiosis up to 3 months after birth. My background is in meiosis. So it's always important for us to analyze how this is happening, but also it was important to know if these cells were like living next to the cells positive for pluripotency. And in fact, we can see here that they're in green positive SOX2, REC8 specific cohesion, and they're like living all happily together. We analyze them across the ages, and we can see, in fact, how they're like going into meiotic prophase. We count them, and we know that they're entering into meiosis here. They drop, but they're still entering later on in life. Again, since as I mentioned, I work in meiosis in the past, we always want to know what is the quality of the eggs, because as you know, if an egg is bad, they could be induced premature ovarian insufficiency. So the expiration date of the ovary comes first. Also, it can increase the risk of aneuploidies, miscarriages, et cetera, et cetera. So there are like 3 processes that we evaluate. Basically, there is a pairing synapsis and recombination. So what are you seeing here are markers of DNA damage and repair, and you are seeing how they repair properly, they synapse, and also they're forming the crossovers. These crossovers are really, really stable, and we can see how each one of the chromosomes of the naked mole rat, they have at least one. And we can count them, they have 1, 2, and we analyze them. And something that is interesting is that this number is always really variable in mouse and human, but in this case it's really, really kind of tight. One point that is important and we are working on is that there is a slightly drop with age, and actually is what we see in human. So now that we know this, the next thing is like, okay, you have really cool pictures, but it's a new species. How do you know that actually what are you seeing is real? So we did, now we have single cell, but this was a bulk RNA-seq with the ovaries, and we were able to detect the different markers. For here you see like pluripotency, you can find your favorite meiosis gene to confirm what we were seeing. So now we know, and then we analyze them and compare them to mouse, where we see that in this case, naked mole rat, they're like present all the way, the pluripotency factors. Meanwhile, in mouse is just like in this really specific time. So now we can say that, okay, we know that there are tons and tons of cells. They have they're positive for pluripotency factors, and they have all the cells living together. The second question that we have is like, okay, so do they kill a lot of cells or not? So what we did to analyze this is like a simple assay. We detect the tunnel assay, clipper space, 3 and 9, et cetera. And what we saw is that, yes, there's a peak in cell death about P15. But even with this situation, the naked mole rat is still having 1 million of germ cells. That is exactly what I mentioned at the beginning with human. So there are a lot, we kill less. So the third question was like, are we putting more cells into the pocket? And this was a tricky one. So let's start and look for markers of cell division. And what we found is that, or not found. Okay, so why we think that they are dividing? Well, we think this because basically what we saw is that, well, let's go to this one. So what we see is that they're like markers of cell division. We have Ki67 and BASA, and we see these lovely clusters in P1, also P5, P8, et cetera, and then going up to P90. We count them, they're still there all the way, a little bit lower here, but still present. And again, since it's a new species, we decided to use different markers. So the next one that we use is phosphohistone H3 that actually show the same results. We count them, same situation. So now the question that we have is like, are these cells that are dividing is just like random germ cells, or are cells that are positive for pluripotency factors? So we did that analysis, and here you can see in red SOX2 and Ki67 in green, and actually the cells are dividing. So this means that basically the cells that we're seeing dividing up to 3 months are like this pluripotency factors positive cells. So I mentioned at the beginning that there's over-optimist and over-pessimist. But at the end of the story, if the cells exist or not, they're doing nothing. So here what we need to know is like, actually, they exist. But they're doing something in adults, because if they're doing nothing, who cares? So we decide to analyze that. And for that, we use 6 months, that is like the earliest age that they can turn into a queen. SUPs, that means here, girls that are in the colony. And XUPs, that basically is a new queen. We analyze them, we count the germ cells, and we didn't see a lot of differences at the beginning, but it was like, okay, it seems like it's going up. So we decided to use the markers that I mentioned before. And what we saw is actually that in the case of the queens, there's a raise in the number of SOX2 positive cells. The only reason to have this is that they're dividing. So we went again to the next step and looked for them and tried to see if they were dividing. And actually, we saw that they were positive for Ki67, phosphohistone H3, and they're also SOX2 positive. There's a little bit in the SUP in 6 months, don't take me wrong, but the number that we have in the queens is way bigger. So the next question is like, okay, if you have them, it's cool, but if they get stuck in there, again, they don't work for us. So what we try to do next is to analyze how far in the development you can push them in vivo. And this is analysis with markers of meiosis. And what we actually see is that in adults, these cells are going into meiosis, but also that we are able to collect them and get oocytes. These eggs are from queens, and new queens and subordinates. And we also saw that A, we can get them, but B, there's difference between them. We can get less in the subordinates compared to the queens, but also they're like smaller, and in the subordinates bigger in the queens. The progression in the in vitro fertility for in vitro maturation is different, and the queens just do better. Also, they're able to progress, and here we have an analysis of how they look across the in vitro maturation. And something really cool is that if you check the times that they have to do this, is actually basically a copy and paste from human. It takes the same time to do the in vitro maturation. Our embryos are not the best, but working on. So the next question is like, if these cells are positive for pluripotency, they should be able to grow in vitro, right? Otherwise, like something sketchy happening there. So what we did is like to develop a system to culture these cells. And actually here you have cells that we start from ovaries at P5, and we culture for 5 days, 10 days, so on, and we see these clusters. They can go further, and in fact we are able to culture them up to 120 days, and you can see that they're like forming these clusters that we call nest, and here you can clearly see how they are forming follicles in vitro. Anyways, this is just showing that we know how to maintain them and push them, but it's not saying that actually they are dividing in vitro. So we use the help of a really easy trick that all of you know really well. So we treat them with EDU, and we can see that there are cells positive for EDU and SOX2 and BASA, but not all of them are positive for EDU, showing that actually they are dividing. So that was really early ovaries. And again, it turns to the question of maybe it's a developmental delay. Who cares? Because what we need is to have them working properly in adults. So what we did is to try to culture ovaries from subordinates and also the neoqueens. Well, first thing, in the case of the subordinates, we have the best amazing fibroblasts, not germ cells. In the case of the queens, what we saw is like a phenotype similar to what we observed in the early ovaries. And also, when we follow with the different markers, we can see that actually our new cells that are positive for these pluripotency factors, positive for germ cells, and positive for EDU. So now we can say that in early and postnatal adult naked morat queens, the ovaries that have the markers, they are dividing in vivo and in vitro, and they can go faster, and you can generate follicles. So, going back to the initial question, like how this little animal is able to do this? Well, now we can say that A, they have a lot. So that's why they have tons and tons of germ cells. B, they kill way less than other species. And C, they're putting more into the pocket. So what are we doing now? The story here is like how this is happening and how we can use it in other species. So this is just a little bit about what are we thinking that is happening. Okay, let's think about the mechanism of aging of the naked morat. So we know that there are absolutely gorgeous genes with genomic stability, oxidative stress, protein homeostasis. They have a really peculiar extracellular matrix that actually seems like it's really important for the ovarian development. And they really go to cell cycle regulation. But this is the basic package. This is go to the dealer, get the car, and the simplest one. But there's some difference, and it seems like the queen had like an extra value. And those extra values are, they have like a slow epigenetic clock, let's say. They have these cells that express the Yamanaka factors, basically. They divide, and they conserve the lineage. And basically, this is what yesterday we hear two of the global experts talking about partial reprogramming. So what we think is that in the case of the naked morat, the queens can be a model for partial reprogramming. And in fact, the cool part about this is that we are following these cells. And now we know they're like doing this in pulses, similar to the model that yesterday mentioned. So we think that one extra level that they can have is that they are this model for partial reprogramming and rejuvenation. So now we are involved in a massive cohort where we are analyzing how the activation changed the epigenetics, how it in the ovary and in different organs, and see what are the mechanisms that are triggering this and regulating from the ovary to the brain and other tissues. So with this, I want to say thank you for all the people in my lab that helped me to develop this, our funding. And with this, I would like to take questions if you have any. Thank you. Or not. What's that? No question.
Are there any questions in the audience? For now, or you're waiting for the panel? Oh, there is one.
So can I ask, you said that these animals are making partial reprogramming, so we can learn on them also how to do reprogramming on humans. It can help us.
I think that there's a magic. So what Dr. Serrano and Ocampo developed in vitro, it seems like this animal is doing by itself. So it seems that it's regulated with the cells, basically with a period, and it's every 30 days. So, but we don't know exactly what is triggering it. So we are embarked right now to try to analyze what is that thing that they actually naturally have to try to see if this is conserving other species. Because if that is real, that means that we kind of skip a lot of situations. Obviously, we are far away to know exactly what is happening, but definitely it's something that we're looking from all the levels, like multiomics, proteomics, and metabolomics. Yeah.
There's one more question here. Yeah.
Great talk. But what is required to make a queen? So what has to happen? So if you were to take away the queen, you have all these young potential queens. How is a queen decided?
Okay, how was established the first queen, we don't really know. But in the colony, there is a queen. And there are like certain girls that are like semi-active, let's say, and you can see certain features. So if the queen dies, one of them will take the place. So they can fight to get that point, but that is something that we don't really care in this point. It's supposed that it's regulated by different aspects, so there's like a little bit of pheromones, touch. So there are like many things involved. We don't have a clear mechanism, but what we know for sure is that as soon as you remove them from the social environment, they trigger. And 4 to 6 weeks later, if you put it with a male, you can start breeding new animals. So it's activating. Something really interesting is that in some of our colonies, we have queens that abdicate. So suddenly, a queen of 16 years old stopped breeding, and there's another girl that takes the place, but they never fight. So what this led us to do with this queen is like, what happened if that queen that is inactive, if you take it out, it turns active again, and it seems that is the case. So that is talking about how intricate and delicate is all the epigenetics in this animal, because basically it's nothing really changing except that. So there's something, it needs to be something that you can turn on and turn off quite fast.
Thank you. I would wrap up. We have one more question. Maybe we will take this one and then move to a next speaker. Yeah, thank you.
Did you say there were two eusocial animals? Two
Yeah.
social mammals, rather? Are
Yeah.
there investigations into the other species as well and what we can learn from it?
Okay, so it's another of the African mole rats, and yes. So we are, so Vera Gorbanova, yeah, you probably know really well, and also Melissa Holmes, we work in collaborations with them, and they have this specific one. So we are like looking if it's the same case for those ovaries. I highly doubt, because I really believe that, and this is me without like a lot of evidence, that we don't have a magic key. The naked mole rat is a glitch in the system with tons of things that protect them from aging and give the opportunity to have babies forever. So, but it's something that we are looking for. Thank you.